In 1950, the US Navy secretly sprayed bacteria over San Francisco, resulting in serious infections and death that they tried to cover up for decades. Back in the 40s and 50s, pregnancy tests involving frogs went wrong when frogs escaped, spreading a deadly fungus that almost wiped out native frogs around the world. These are disturbing experiments in nature gone wrong. Biosphere 2 was set up as a 3-acre capsule to mimic Earth's ecosystems. Rainforest, ocean, desert, and farms. It was all sealed shut on September 26, 1991, with 8 scientists locked in for 2 years. By month 16, oxygen levels had fallen from about 21% to just 14%, roughly the equivalent of living at 13,000 feet. This wasn't good. Headaches, fatigue, insomnia, and mental fog was a daily struggle. One participant, Mark Nelson, couldn't even do simple math. Kind of like me. Scientists from Columbia University, Jeff Severingus and Wallace Broker, later discovered that the soil, packed with peat and compost, had microbes in it that consumed oxygen and produced CO2. But much of it was absorbed by calcium in the concrete walls, locking away CO2 and oxygen in solid form. Add blocked sunlight, poor plant growth, dying bees, and runaway ants, and the ecosystem was clearly out of balance. Even with daily food production that supplied about 83% of their diet, the crew lost an average of 16% body weight during the first 6 months. And things got tense between the scientists too. Factions started to form. Enough that participant Jane Pointer said that living there was, quote, cult-like. By the end of the mission on September 26, 1993, the crew finally came out. Thinner, exhausted, but alive. Suffice to say though, things hadn't gone smoothly. In September of 1950, the US Navy carried out a secret biological warfare test on civilians in San Francisco. They loaded a Navy ship with massive tanks of bacteria and used hoses to spray a cloud of microbes into the air near the coastline. That sounds like something Lex Luthor would do, but the goal was to test how easy it would be to infect a major US city with biological weapons. It still sounds like something Lex Luthor would do. Now, they weren't actually trying to just kill people outright. The bacteria they used was believed to be harmless at the time, but they weren't. After the spraying, hospital staff at Stanford started seeing strange infections. 11 people developed serious urinary tract infections, and one man, Edward Nevin, died after Serratia infected his heart valve. It was the first recorded outbreak of Serratia in the area. The doctors couldn't explain how such a rare bacteria had suddenly shown up. The Navy kept the test a secret until the 70s, when documents were released during a Senate investigation into Cold War experiments. Turned out similar tests had been done in other cities as well. Operation Sea Spray is now one of the most infamous examples of government experiments that went way over the line, which is putting it lightly. Back in the 1940s and 50s, doctors used African clawed frogs for pregnancy tests because they had a weird but useful reaction to pregnant women's urine. Here's how it worked. Urine samples were sent to labs where technicians would inject a small amount into a female frog's hind leg. Then they put the frog back in its tank and checked the next morning for frog eggs in the water. If the frog had laid eggs, that meant the woman was pregnant. Pregnancy hormone called human chorionic gonadotropin triggered the frog to ovulate. Labs shipped thousands of these frogs all over the world. When the frogs got loose or were released into ponds and streams, things got kind of bad. African clawed frogs are pretty tough and they're invasive. They also carried a deadly fungus which didn't hurt them but killed off native frogs everywhere they spread. This fungus attacks amphibian skin, making it almost impossible for them to regulate water and electrolytes, which often leads to death. So frogs started dying off in places like California and Australia. Some species actually came pretty close to extinction. So eventually they put a stop to these tests. In 1962, scientists at the University of Oklahoma decided to test how LSD affects elephants. They wanted to see if it could trigger what's called the must, which is a natural period when male elephants get more aggressive, unpredictable, and start reproducing more. To do this, they gave an Asian elephant named Tusco an enormous dose about 3,000 times what a typical human dose would be. Well, within minutes, Tusko collapsed and went into seizures. The team tried to save him with sedatives and antipsychotics, but an hour and 40 minutes later, he died. Scientists later realized the dose was way too high. And some think the sedatives may have made things worse as well. When other researchers gave elephants smaller doses later on, those elephants survived just fine. Obviously though, this was a pretty disgusting, ill-conceived experiment. On October 13, 1947, US military and General Electric teamed up for a pretty insane experiment. They flew into Hurricane King, a massive storm moving out to sea, and dropped a bunch of dry ice into its clouds. They hoped that the dry ice would create ice crystals, collapse the eye wall, and weaken the storm. At first, it looked pretty promising too. The shape and movement of the storm's clouds looked different after they dropped the dry ice into it. But then the hurricane suddenly veered west, made landfall near Savannah, Georgia, and tore through coastal areas with 85-mile-an-hour winds, flooding streets, wrecking homes, and killing at least one person. The public accused the project of causing this disaster, forcing General Electric and the military to pull the plug. It turned out they probably didn't cause the turn, but the experiment left such a bad reputation that projects like this vanished for over a decade. In secret trials during World War II, the US and New Zealand tested whether explosives could trigger tsunamis. Led by engineer Thomas Leach, the project used thousands of small underwater explosions off Auckland's coast, where test blasts formed mini waves. They found that chaining together tons of explosives about 8 kilometers offshore could, at least in theory, create a wave big enough to flood enemy shorelines. They never tested it at full scale, thankfully, but the results showed that it could work. Officials called it a tsunami bomb, and some thought it could be as destructive as the atomic bomb, just without the radiation. There was real military interest in this for a while, but by the time they were getting close to something usable, the US dropped atomic bombs on Japan, and this idea got shelved. The project stayed classified until the 90s, when declassified papers confirmed that yes, the testing was real, and no, it wasn't just some Cold War myth. In May of 1955, the US government dropped 300,000 mosquitoes over Georgia to test how well these bugs could spread and survive after being released from planes. This was part of Operation Big Buzz, and it worked well enough that the government went on to launch even bigger tests called Operation Dropkick, Operation Gridiron, and Operation Mayday, spreading mosquitoes over parts of the South. They wanted to eventually use mosquitoes carrying diseases like malaria or yellow fever as biological weapons to weaken enemies during wartime. Just nuts. Years before this, though, during World War II, US officials wanted better malaria treatments, and they needed human test subjects to see how effective they were. So they turned to Stateville Penitentiary in Illinois, offering inmates reduced sentences if they volunteered to be bitten by infected mosquitoes. 10 bites a day, to be exact. Over 400 prisoners signed up, and the results were pretty awful. Inmates suffered fevers as high as 106 degrees, and some anti-malaria treatments caused serious side effects like heart attacks. That's as serious as a heart attack. These experiments went on for 30 years too, often without proper consent or care. In 1971, microbiologist Ananda Chakrabarty at General Electric engineered a strain of bacteria that could break down crude oil faster than any natural bacteria. He combined plasmids, which are basically little loops of DNA that bacteria use to share useful traits, combined them with 4 oil-eating bacteria into one superbug, hoping to use it for cleaning up oil spills. Well, it worked in the lab, and in 1980 this bacteria became the first genetically modified organism to be patented in the United States. The idea was to throw this into an oil slick and let it eat through the mess. That's what eventually happened. After the Deepwater Horizon spill in 2010, natural oil-eating microbes exploded in number and helped digest millions of gallons of crude, but that was nature's own doing. When humans start releasing engineered bacteria, things can get dicey. There are concerns about disrupting microbial ecosystems, depleting oxygen in the water, or creating unexpected chemical byproducts. So, yeah, that all sounds pretty bad. Bacteria that eat oil sounds like a miracle solution, and in some ways it is. But at the same time though, tweaking even the smallest parts of nature can have ripple effects that we still don't fully understand. Bringing animals back to the wild sounds great too, but it's a lot harder than it seems. In the 1960s, conservationists tried reintroducing black-footed ferrets in the US, which were on the verge of extinction. They bred ferrets in captivity and then released them, but most of them died, either from diseases, habitat loss, or lack of food. The ferrets were competing with native predators as well. Another example is the Arabian oryx. Even though conservationists released captive bred oryx into the desert, poaching kept their numbers low. So saving a species isn't just about putting an animal back into the wild. Without the right food, safe spaces, protection, it can turn into a long, painful struggle. In 1953, British scientist Bernard Kettlewell released light and dark-colored peppered moths in the woodlands of Birmingham and Dorset to test whether pollution was helping dart moths survive better by hiding them against soot-covered trees. He marked and tracked hundreds of moths and watched birds hunt them down. The data showed dart moths did have an advantage in polluted areas, while lighter ones did better in clean forests. Natural selection right in front of your eyes. But later on, people poked holes in it. Moths don't usually rest on tree trunks, which is exactly where Kettlewell placed them. The photos used to illustrate his findings were staged as well. In 2002, journalist Judith Hooper called the whole thing into question in her book Of Moths and Men, writing that the science was sloppy and biased. But researcher Michael Majerus spent 6 years between 2001 and 2007 redoing the study in a more natural setting. He found the same result though. Dart moths were eaten more often in clean environments. So the conclusion still stands, but the story's more complicated than the version that showed up in high school science books. I'm your host, James. I'll catch you, yes, you specifically, in the next video.